Welcome students. Today we are going to be studying about the microorganisms in air. The title of the paper is Microbiology and Plant Pathology. The unit is Microbiology of Air and Soil and Water. The name of the module is Occurrence of Microorganisms in Air. I'm Lynette Fernandez, Associate Professor from PES College. The outline of this paper is microbiological content of in air, physical environmental stresses, enumeration of microorganisms in air, significance of microorganisms in air, control of airborne microorganisms. What are we going to learn from this? Why is why air is not a natural medium for microbial growth? We will be understanding the techniques for microbial analysis of air, and also we will be studying about the significance of microorganisms in air. The microorganisms present in air are studied under the heading aeromicrobiology. It is a study under it is an applied aspect of aeromicrobiology. In air, there are various kinds of microorganisms. They might be present in the form of vegetative cells and spores. So vegetative cells and spores of bacteria are present, fungi and algae, viruses, protozoan seeds, spores of pteridophytes, and pollen grains from angiosperms. Very commonly, we find the spores of Bacillus, Clostridium, ascospores of yeast, Conidia of Cladosporium, Penicillium, Aspergillus, etc. Even though these microorganisms are present in the air, they are not the natural inhabitants of air. They occur in the form of colloidal systems or the so-called bioaerosols. They are usually found bound to dust particles or liquid droplets. They are not indigenous to air because they are not the natural flora of air. They are swept into the air by air currents. They are swept into the atmosphere by the air currents. Common sources from where they are swept into the atmosphere are water, soil and plant and animal surfaces, and human beings. In the case of man and human beings, a large amount of organic wastes which are disseminated in the air contribute to the microbial flora of air. While coughing, talking, laughing, or sneezing, large amount of droplets from the nasal passages or from the oral passages are given out by men into the air. These droplets contain large amount of microorganisms from within. Air does not support the growth and reproduction of microbes. Even though these microorganisms have landed in the air, they will not be able to survive in the air for a very long time. So, they are unable to grow because they do not have, there is no enough moisture in the atmosphere. The nutrients are not present in the air and they do not have anchor for the microorganisms. Air is only a medium of transport for the microorganisms. Once the microorganisms reach the atmospheric column, they are transported from one place to another place. Besides this, the microorganisms have to bear a lot of stresses, physical environmental stresses, once they are present in the atmospheric column. One of which is desiccation, which is a primary environmental stress which they have to face. And this will decide how long they will be able to survive in the atmosphere. Next is the temperature. In the case of the temperature, the organisms are unable to survive at high temperatures because their proteins get desiccated. Their proteins get denatured. Some of the stresses which they have to face are the physical environmental stresses, one of which is desiccation. Desiccation will decide how long the organism will be able to survive in the atmospheric column. Temperature also is an important environmental stress. High temperatures can bring about the denaturation of proteins. Very low temperatures forms ice crystals. Some organisms are able to withstand high humidity conditions. Some organisms are able to withstand only low humidity conditions. Air currents also is an important environmental stress for them. Radiations, they are exposed to various kinds of radiations in the atmosphere coming from the sun, and their DNA can get denatured. Microflora of the air can be studied under two heads: outdoor microflora and indoor microflora. Both are important. In the case of our outdoor microflora, it deals with the study of microorganisms which are present in the open atmospheres over the land and sea. In the case of indoor microflora, it deals with the organisms present in the atmosphere or in the air of hospitals, houses, and library. In the hospitals, the air may contain a lot of pathogens which can cause a lot of disease. In the houses, even though they might appear to be clean, they contain a lot of spores. Library contains lots of books, so there are many cellulose degrading organisms. Significance of microorganisms in air. It is very important to study about the microorganisms present in air because they are responsible for bringing about a lot of damages. They are responsible for biodeterioration of storage materials, equipment, library materials, archives, wall paintings, etc. They also cause the spoilage of foods and fermentation products. They are also responsible for animal and plant diseases, which is very important as far as agriculture is concerned. Fungal spores and pollen grains cause a lot of allergies in men. Many of the bacteria, viruses, fungi present in the air are responsible for airborne diseases in humans. Microorganisms in air can be studied by using different techniques: impingement in liquids, impingement on solids, and membrane filters. Impingement in liquids. Here, the air is drawn through a very small opening in a tube and it is bubbled through the liquid. The organisms get trapped in this liquid medium, and aliquots of this liquid medium are subsequently plated or cultured to determine the microbial content present in the air. Impingement on solids. Here the microorganisms are collected directly on the solid surface of agar medium or on the filter disc. Colonies will develop subsequently where the organism impinges, and that is how it can be studied. A very common technique here is the settling plate technique. Here, the petri dishes containing the media are exposed for a certain duration of time to the atmosphere. Fungal or bacterial spores will settle on the media, and ultimately they will start developing colonies once the petri plate is incubated. This will allow one to know about the different types of microorganisms that might be present in air. But here, the volume of air is not known. So there are other techniques which also can be used, such as sieve and plate type devices. In and sieve and slit type devices, measured volume of air is sampled. In slit sampler, the air passes through a slit which is kept above a petri plate containing the agar medium, and the petri plate is rotated so that the microorganisms are evenly distributed on the agar plate, and later on it is incubated. In the case of the sieve device, there is a metallic sieve containing having very small openings which is placed over the agar surface. The air is allowed to pass through these small holes in the sieve and reach the agar surface along with the microorganisms. Membrane filters. In the case of membrane filters, a filter assembly is used on which a membrane is placed aseptically. Air is allowed to pass through this membrane, and the microorganisms which are present in the air will get impinged on the membrane. The membrane is removed aseptically from the filter assembly and it is placed on a disc of absorbent paper saturated with appropriate medium and it is incubated. It is important to control the microorganisms present in the air, especially indoors. This is done through various methods. One is by directly controlling the dust, ultraviolet radiations, bactericidal vapors, laminar airflow systems. Dust control. Here, the dust in the room has to be controlled while room cleaning operations are being carried out. And this can be done by oiling the floors, bedclothes, and other textiles. Sometimes a dry vacuum pickup of dust can also be followed. And this can be followed by the disinfectant and detergent solution cleaning. Ultraviolet radiations. Ultraviolet radiations are very harmful to the cells. They cause damage to the DNA. So there are germicidal lamps which are often used in rooms which radiate ultraviolet radiations of wavelengths in the range of 250 to 260 nanometers. This can kill the microorganisms if they come in contact with these radiations. Now, this can be done in two ways, either by using direct radiations or indirect radiations. Direct radiations is usually carried out in rooms which are not occupied or aseptic filling rooms of pharmaceutical preparations. In the case of indirect radiations, these are carried out in occupied rooms, offices, hospital wards, etc., where the radiations will not come in direct contact with the people. Another method is bactericidal vapors. Chemical substances are vaporized or sprayed into the air of a room. These bactericidal vapors are dangerous for the organisms and can kill them. So, such vapors of triethylene glycol, lactic acid are often used in hospital rooms. And they should be effective at normal room temperatures and humidity. They should not be toxic to people or they should not be corrosive and they should be colorless as much as possible. Laminar airflow system. In the case of laminar airflow system, the air is allowed to pass through high efficiency particulate air filters of cellulose acetate. The particles as small as zero point 0.3 μm are removed by this filter system and thus helps in cleaning up the air which enters a room from any microbial contamination. There are other ways through which the air can be kept clean. One of the very effective way in controlling airborne disease indoors is natural or mechanical ventilation of rooms. A well ventilated room will be free from any airborne diseases. Thank you.